Imagine a pain so severe that, well, just a feather brushing against your cheek feels like a blowtorch.
Yeah, it's a completely terrifying concept.
Right, it's this sharp, shocking agony. And it doesn't just hurt, it completely drains the life out of you.
It really does. It's exhausting.
Exactly. Now, for you listening, I want you to imagine that alongside that intense physical agony, a really heavy, dark depression settles over your mind.
Which is incredibly common, unfortunately.
Yeah, if you've ever experienced severe chronic pain or, watched a loved one go through it, you know exactly what I'm talking about. The physical suffering is rarely just this isolated event.
No, almost never.
Right. It almost always brings this intense anxiety and a crushing sense of despair along for the ride. And for decades, the standard medical assumption has been that the depression is simply, well, a psychological reaction to being in pain.
Right. It was seen as purely secondary.
I mean, it makes intuitive sense, right? You hurt, therefore you are sad. Okay, let's unpack this. Because a groundbreaking 2026 paper published in the journal Neuron by Kai and colleagues completely upends that assumption.
It really is a paradigm-shifting piece of research.
It reveals that the physical flinching and the emotional despair are actually the exact same biological malfunction, which is just wild to me.
It is wild. And it's so important because if you assume the depression is just a secondary reaction, your whole treatment strategy is inherently flawed.
Oh, because you're treating the wrong thing.
Exactly. This is exactly why traditional painkillers so often fail people with complex long-term conditions. I mean, if you prescribe a pill that only numbs a peripheral nerve, in the face or the back, you are entirely ignoring the comorbid emotional piece of the puzzle.
You're putting a Band-Aid on half the problem.
Right. What this new research shows us is that the emotional despair isn't just a side effect. It is structurally baked into the exact same brain circuitry.
Okay, so where in the brain are we talking about here?
So today we are looking at a very specific neural highway. It starts deep in the midbrain in a region called the ventral tegmental area, or VTA.
The VTA, okay.
Right, and it travels all the way up to the front of the brain, basically right behind your forehead, to the orbitofrontal cortex, which we'll just call the OFC.
Okay, so we have this VTA to OFC highway. And to figure out what's actually happening on that road during chronic pain, the researchers obviously couldn't just ask people how they were feeling.
No, human studies are too messy for this kind of precise cellular mapping.
Right. So they needed a highly controlled model, which means they turned to mice. And they were trying to replicate a very specific, devastating human condition, right?
Yes. They modeled a condition called trigeminal neuralgia.
Trigeminal neuralgia. What does that actually look like in a person?
In humans, it involves extreme shock-like facial pain. It is incredibly severe. In fact, historically, it's been referred to as the suicide disease.
Oh, wow. That's a horrible name.
It is, but it speaks to the fact that the emotional toll it takes on patients is just staggering. The pain is that unbearable.
So how do you even replicate that in a mouse?
To study this in the lab, they used a model called CION. That stands for chronic constriction of the infraorbital nerve.
Chronic constriction, so they literally pinch the nerve.
Basically, yes. They perform a minor procedure to constrict a specific satial nerve, which effectively mimics that severe, persistent nerve pain you see in human trigeminal neuralgia.
Okay, so once that CION model is set up, how do you actually measure a mouse's physical pain versus its emotional state? Because obviously a mouse can't, fill out a depression questionnaire.
Right, they can't tell you they're sad. That's where behavioral science comes in, and the researchers had to be really meticulous here.
Because they have to separate the two things.
Exactly. So to measure the physical pain, they looked for something called mechanical allodynia.
Allodynia. That's a great word. Let's break that down for the listener.
Allodynia is that phenomenon you mentioned earlier in the intro, when a completely benign, non-painful touch suddenly registers as agonizing pain.
Like the feather feeling like a blowtorch.
Precisely. To test this, they use tiny, highly calibrated fibers called von Frey filaments.
Von Frey filaments?
Yeah, they're like microscopic little hairs.
Yeah.
They gently press these filaments against the mouse's whiskers. Now, a healthy mouse wouldn't care at all. It's just a light touch.
Right. It's just a tickle.
Right. But these mice, with a nerve constriction, they would flinch and pull away at the absolute slightest touch. That proves the severe physical pain.
Got it. So that gives us a really clear metric for the physical sensitivity. But what about the despair, the anxiodepression, as the paper calls it?
For that, they observe how the animals cope with their environment. So they use a few well-established tests. One is an elevated plus maze to measure anxiety.
Okay, an elevated maze. What are they looking for there?
They're basically watching to see if the mice bravely explore the open, exposed arms of the maze, or if they cower in the enclosed, dark spaces. Highly anxious mice will hide.
That makes sense. What about the depression side of it?
They use a tail suspension test. It measures how quickly a mouse gives up struggling when placed in an uncomfortable but completely safe situation.
Like hanging them by their tail.
Exactly. Giving up quickly, just sort of going limp, is a classic, widely accepted indicator of depressive-like despair in animal models.
Okay, so the researchers have the physical pain isolated with the whisker flinching, and they have the emotional despair isolated with the hiding and giving up.
Right. Two distinct measurements.
Now comes the really fascinating part. They want to look directly at that neural highway, the VTA to OFC circuit, while this is all happening. And to do that, they use some truly mind-bending technology, right?
Oh, the technology here is astonishing. They utilized genetically encoded fluorescent sensors called GrabDay.
GrabDay. Okay, what does that actually do?
They essentially injected a specialized protein into the brain that is designed to literally light up, it glows green, the exact millisecond that dopamine binds to it.
Wait, they literally make the brain glow in the dark?
Yes. And then they threaded microscopic fiber optic cables into the brains of the living, awake mice, right into the orbitofrontal cortex.
So they are literally watching a live video feed of the brain glowing green every time a wave of dopamine hits the frontal lobe. That is sci-fi level stuff.
It really is. And what they saw when they looked through those fiber optics is the core discovery that sets up everything else we're going to talk about today.
Okay, lay it on me.
They found that the chronic nerve pain creates a profound hypodopaminergic state.
Hypodopaminergic, meaning low dopamine.
Exactly. It was a massive, sustained drop in dopamine levels in the OFC.
Okay, let me try to use some visual memory here to visualize this for the listener. Let's compare that deep midbrain structure, the VTA, to a high-powered dopamine manufacturing plant.
I like that, yes.
Its whole job is to produce this chemical. And the OFC, right behind your forehead, is the executive control room that absolutely needs a steady shipment of that dopamine to function properly.
Right. It needs those regular deliveries to keep your mood and sensory processing stable.
So the researchers are sitting in the control room, looking at the delivery chutes, and the shipments have just completely stopped. The factory has shut down.
That is a perfect way to look at it. The pain signal essentially starves the executive part of the brain of dopamine.
But seeing that immediately makes me wonder if the factory shutting down is the root cause of all these terrible symptoms, you know, the flinching, the hiding, the despair. Why can't we just kickstart the factory?
What do you mean?
I mean, if you know the dopamine plant is offline, can't you just give it a jolt, wake it back up to fix the problem?
That is the exact logical leap the researchers made next. And because they are working in a modern neuroscience lab, they didn't just use a generic drug to try and wake it up. They used lasers.
Lasers, of course they did.
It's a technique called optogenetics. They went back to the dopamine neurons in that VTA factory and genetically tweaked them to express a protein called CHR2.
CHR2?
Yeah, it basically makes those specific neurons extremely sensitive to light. Then they pulsed a highly specific wavelength of blue light directly onto those neurons.
They literally jump-started the brain's factory with a laser beam.
They did. By flashing this blue light 30 times a second, what we call 30 hertz phasic bursts, they forced the dopamine neurons to fire.
Like revving an engine.
Exactly. It forcefully pumped dopamine back up the highway into the OFC. And the results were profound.
Yeah.
The moment they restored that dopamine flow, it completely rescued the animal.
Wait, rescued in what way?
The physical pain, the flinching at the von Frey filaments we talk about, it vanished. And simultaneously, the depressive behaviors vanished too. The mice started exploring the open maze again. They didn't give up on the tail suspension test.
Wow. Okay, so let me do a quick recap here to make sure I'm wrapping my head around this first major piece of the puzzle.
Go for it.
Bullet 1, the VTA-OFC dopamine circuit is deeply suppressed by chronic pain. It's a factory shutdown. Bullet 2, this low dopamine state is responsible for both the sensory pain and the emotional pain.
Right.
And bullet 3, forcefully reactivating this pathway cures both problems simultaneously. So the brain isn't dealing with two separate diseases. It's a single dopamine drought causing both the agony and the depression.
Exactly.
For you listening, have you ever noticed your own physical aches and pains spiking on a day when you're just incredibly emotionally drained?
That's a great question to think about.
Right, because for years, doctors might have said you were just hyperfixating on your pain because you were sad. But this research suggests it's a structural reality. Your emotional resilience and your physical pain tolerance are drawing from the exact same dopamine well.
It totally validates the experience of a bad day.
Yeah.
But this actually brings us to a really tricky biological mystery.
Okay, what's the mystery?
If one single surge of dopamine from the midbrain travels up to the OFC and fixes two very distinct problems, mood and physical pain, the brain has to be splitting that signal somehow, right?
Oh, I see. The dopamine must be hitting a fork in the road.
Right. How does the executive control room sort out which part of the dopamine is for mood and which part is for physical pain relief?
That is the million-dollar question. So how does it sort it out?
To answer it, we have to look really closely at the receptors. Dopamine doesn't just wash over the brain randomly. It's like a key that only fits into very specific microscopic locks located on the outside of other neurons.
Okay, so the receptors are the locks.
Exactly. The researchers mapped out the OFC and found two distinct types of locks waiting for that dopamine. The first type they focused on are called D1-like receptors, or just D1.
Okay, so the D1 locks, where are they located, and what happens when the dopamine key turns in those locks?
Well, these D1 receptors sit mainly on excitatory pyramidal neurons in what we call layer V of the cortex.
Layer V, got it.
And chemically, the D1 receptor is what we call a G-coupled receptor.
G's coupled. Okay, we need a mnemonic for that. Let's tell the listener to think as for stimulatory.
That's perfect. Yes, as for stimulatory. When dopamine binds to it, it activates an enzyme called adenylate cyclase.
Adenylate cyclase. And what does that do?
It ramps up the production of a messenger molecule called CAN-MP inside the cell. Basically, this opens up the electrical floodgates, causing the neuron to fire off its own signal.
So dopamine hits the D1 lock, the CAN-P goes up, and it essentially shouts, go, to the cell. It stimulates it to fire its message further down the line.
Exactly. Now, the researchers wanted to know what only this D1 signal does. So using those targeted optogenetic tools, they activated just the D1 excitatory neurons in the OFC, completely ignoring the rest of the brain.
Here is where it gets really interesting. What happened when they hit only D1?
When they activated the D1 neurons, it completely cured the emotional despair. The anxiety vanished, the depressive hiding stopped.
Okay.
But, and this is crucial, it did absolutely nothing for the physical pain.
Wait, really? Nothing at all?
Nothing. The mice were perfectly happy, they were exploring, but they still flinched in absolute agony when their whiskers were touched with the filaments.
That is wild. The mood is completely fixed, but the physical pain is entirely untouched. So the signal physically split.
It did.
To go back to my earlier analogy, it's like a train leaving the VTA station loaded with dopamine. When it hits the OFC switchyard, the tracks diverge. The D1 train is specifically on the emotional healing track.
Yeah, that's exactly right.
But where is that track actually going? Where does the D1 neuron send its message to fix the depression?
They traced the axons, those long wire-like outputs of the D1 neurons, and found they travel all the way down to a region called the LDTg.
LDTg, what does that stand for?
It stands for the laterodorsal tegmental nucleus. It's a deep brain region that is entirely dedicated to processing emotions and motivation. It bypasses the sensory pain centers completely.
So let's recap this second segment. Bullet 1. D1 receptors are G's coupled, meaning stimulatory. Bullet 2. Activating D1 neurons fixes mood, but not physical pain at all. And bullet 3. D1 neurons route their emotional healing signals to a completely separate destination, the LDTg.
Perfect summary.
So for you listening, what stands out to you about the fact that your brain dedicates an entirely separate physical highway just to process the emotional suffering of pain?
It really implies that from an evolutionary standpoint, the emotional distress of an injury is just as biologically critical as the tissue damage itself.
The brain literally needs you to feel despair so you rest, heal, and, avoid whatever hurt you in the first place.
Exactly. It's a survival mechanism.
Okay, so if the D1 track is exclusively handling the emotional baggage, it naturally stands to reason there must be another track handling the physical pain.
Yes, and that brings us to the second set of locks in the OFC. The D2-like receptors, or D2.
The D2 locks, where do we find these?
These are located in slightly different layers of the cortex, primarily layers 2 and 3. And chemically, they act as the exact opposite of the D1 receptors. They are G-coupled.
Okay, so another mnemonic for the listener. If Kai's was for stimulatory, let's think I for inhibitory.
Exactly. Guy-coupled means they decrease CCAMP levels. Where D1 is stimulatory, the D2 receptors are inhibitory.
Wait a minute, hold on. I need to stop you there because I'm spotting a logical flaw here. That feels fundamentally backwards.
How so?
Well, if D2 is an inhibitory lock, if it quiets the cell down and stops it from firing, how does turning it on relieve pain? Wouldn't inhibiting the brain's pain relief center just make the suffering much worse?
It's a brilliant observation, and it's the exact paradox the researchers had to unravel. To understand it, you have to look at what the D2 receptor is actually inhibiting.
Okay, what is it inhibiting?
You see, the brain isn't just a chaotic storm of electricity. It relies heavily on permanent stop sign neurons. We call them GABAergic inhibitory interneurons.
GABAergic interneurons, so their job is to stop things.
Right. Their entire job is to constantly pump the brakes and suppress the surrounding brain tissue, keeping things calm. The researchers found that these D2 receptors are heavily concentrated right on top of these specific stop sign interneurons.
Okay, I think I see where this is going.
When the dopamine train arrives from the midbrain and the key fits into the D2 lock on that stop sign cell, the inhibitory mechanism turns the stop sign off.
It's a double negative.
Exactly. In neurobiology, we call this disinhibition.
Okay, let me use an analogy here. It's exactly like taking your foot off the brake pedal of a car. The pain relief signal wants to go, but you're holding it back with the brake. The dopamine hitting the D2 receptor is the act of removing your foot from the brake so the car can finally roll forward.
That is the perfect analogy. By shutting down those local GABAergic stop signs, the surrounding excitatory neurons, the ones actually responsible for sending the physical pain release signal, are suddenly freed from their restraints. They light up and fire their signal.
That is just an incredibly elegant system. The brain uses a double negative, this disinhibition, to create relief. So once that brake is lifted and the pain relief neurons are free to fire, where does that second track go? Where does the D2 train end up?
It projects to a completely different destination than the emotional D1 train. The D2 signal travels straight down to the BLA.
To BLA.
The basolateral amygdaloid nucleus, basically a specific part of the amygdala.
Oh, the amygdala. We know that one. That's famously known as the brain's threat processing center.
Right. And in this specific pathway, when the D2 signal arrives at the BLA, it powerfully suppresses the physical pain, the allodynia, without altering the animal's mood whatsoever.
That is staggering. So just to do our third mini recap here. Bullet 1, D2 receptors are G-coupled, meaning inhibitory. Bullet 2, they relieve pain through disinhibiting the inhibitors, like taking your foot off the brake.
Yes.
And bullet 3, the D2 pain relief signal is routed specifically to the BLA and the amygdala. 2 totally separate locks, 2 separate tracks, 1 master key.
You've got perfect.
So I have a question for you listening right now. Can you think of other systems in life, or even your own personal habits, where removing a negative restraint is actually more powerful than adding a positive force?
That's a really interesting way to frame disinhibition in a real-world context. Sometimes you don't need more gas, you just need to let go of the brake.
Exactly. But as much as I love a good, tightly controlled mouse study, I have to ask the skeptic's question here.
I figured you might.
We are talking about a very specific model of facial nerve damage in rodents, the CI-ON model. Are we sure this isn't just a quirky reaction to this one specific injury? Does this intricate D1 and D2 splitting rule apply to other kinds of suffering?
The researchers asked that exact same question. They wanted to know if they had discovered a universal rule of the brain, rather than just a quirk of facial pain.
So what did they do?
They ran the exact same tests using completely different laboratory models. First, they used the LH model, which stands for learned helplessness.
Learned helplessness. What does that model do?
It creates a state of pure depression without any physical pain at all, simply by exposing mice to inescapable stressors.
Okay, so pure depression, no pain.
Right. Then, they used another model called the CFA model, or complete Frohn's adjuvant. This involves an injection that causes localized inflammatory pain in the paw.
So that's pure physical inflammatory pain without the severe nerve damage of the facial model.
Exactly. And they wanted to see, does the map hold up? Did D1 still control mood and D2 still control pain in these entirely different scenarios?
Did it?
Unbelievably well. When they activated the D1 pathway in the purely depressed learned helplessness mice, it completely reversed their despair. But it did nothing for the mice with the inflamed paw.
Wow.
And conversely, when they activated the D2 pathway, it cured the CFA paw pain instantly, but left the depressed mice entirely unaffected.
It wasn't a quirk.
No, it wasn't. They uncovered a universal structural map for how the brain processes these two distinct forms of suffering.
Okay, that proves it in the lab across multiple models. But what about humans? I mean, a mouse is one thing, but human consciousness, our experience of pain and depression is incredibly complex. Do we have any evidence that our brains are using this exact same VTA to OFC dopamine sorting hub?
We absolutely do, and the clinical correlations are really what make this paper so vital for the future of medicine.
Okay, let's hear the human proof.
Let's look back at humans who suffer from that severe facial pain, trigeminal neuralgia. When doctors analyze their spinal fluid, they routinely find abnormally low levels of a chemical called HVA.
HVA, what is that?
It's homovanillic acid. It is a direct metabolic byproduct of dopamine breakdown. So low HVA means low dopamine.
Oh, wow. So this strongly suggests that human chronic pain patients are living in the exact same dopamine drought that the researchers saw glowing green in the mice.
Exactly. The factory is shutting down in humans, too.
That is incredible.
But perhaps the most compelling evidence comes from a completely different branch of medicine, Parkinson's disease.
Parkinson's, really. When we think of Parkinson's, we usually think of the motor symptoms, the tremors, the difficulty walking.
Right, because Parkinson's is famously characterized by the massive progressive death of dopamine-producing neurons in the midbrain. But if you look closely at the non-motor symptoms, Parkinson's patients have astronomically high rates of severe, unexplained chronic pain, as well as deep clinical depression.
Wait, because their dopamine factory is dying off? The exact circuits we've been talking about today are starving.
Yes, the VTA to OFC pathway is losing its fuel.
Okay, wait a minute. My mind is kind of racing here. If their pain is caused by a lack of dopamine hitting those specific D2 receptors, what happens when doctors give them medication for their tremors? Because Parkinson's drugs are basically designed to replace dopamine, right?
This is the massive clinical payoff of this whole deep dive. Drugs designed to treat Parkinson's tremors, like L-DOP, which the brain converts directly into dopamine, or a drug called pramipexole.
Pramipexole, what does that one do?
It's a specific D2 agonist. It basically acts exactly like a master key for those D2 locks we talked about.
Oh my God.
Yeah. These movement disorder drugs are actually astonishingly effective at treating severe neuropathic pain in humans.
That is just mind-blowing. So a doctor gives a Parkinson's patient a pill to help them walk smoothly. And by pure biochemical accident, that pill travels up to the OFC, turns the key in the D2 lock, takes the foot off the brain's internal brake pedal through disinhibition, and cures their chronic pain.
That is exactly what is happening. It perfectly validates the entire D1 and D2 map that Kai and her team spent years uncovering in the lab. The biology translates flawlessly from the glowing fiber optics in the mice all the way to human patients in the clinic.
I'm just marveling at how interconnected all of this is, because historically our modern medical system treats physical pain and depression as two completely alienated domains.
Oh, completely separate silos.
Right. If you have chronic back pain and depression, you go to a pain management specialist for one and a psychiatrist for the other. The pain doctor gives you an analgesic to numb the nerve, and the psychiatrist gives you an SSRI for your mood. We treat them as two totally different problems happening to the same person.
And that siloed approach is exactly what this research challenges. We now have the blueprint showing that they are, very often, born from the exact same point of origin.
The dopamine factory shuts down.
Right, and that single shortage travels up the highway and simply takes two different exits in the frontal lobe, the D1 exit for depression, and the D2 exit for pain.
It really is a massive shift in how we understand suffering. If chronic physical pain and emotional despair are literally just two sides of the exact same biological coin, simply taking parallel tracks out of the same brain region, it fundamentally changes how we need to view people living with chronic illness.
It has to change our view.
For decades, medicine and society in general has had a terrible habit of dismissing the emotional struggles of pain patients, telling them that their depression is just a secondary reaction. Or even worse, implying that their amplified pain is just in their head because they are feeling down.
It's an incredibly isolating, frustrating experience for patients to hear that. But today, we've seen the literal architecture of their experience. We can no longer tell patients that.
Right, because the despair is structurally, biochemically tied to the physical pain. It is unavoidable.
Exactly. So I want to leave you listening with a final thought to mull over.
Go ahead.
If the physical agony and the emotional despair are scientifically proven to be the exact same chemical malfunction, how must our empathy and how must our medical system evolve to treat the entire circuit rather than just endlessly trying to fix one broken branch of it?
That is a powerful question and an essential one to walk away with today. The blueprint for human suffering, and more importantly, human relief, is right there in the biology. It's just up to us to start reading it correctly. Thank you so much for joining us on this deep dive. Keep questioning the assumptions about the world around you. Keep looking for the hidden connections that aren't obvious at first glance. And remember that sometimes the pain and the sadness are just a factory waiting for the lights to be turned back on.